Hi everyone, I'm Kevin Rychel, and I'm the new leader of Genome Analytics at the CFB. So most groups at the CFB can sort of fit into this design, build, test, learn loop. And we at Genome Analytics are the learn group. And you'll notice in this diagram that data is at the center. And that's because data is the core of everything we do throughout the loop. But here in Genome Analytics, it's especially true because our goal is to learn from biological data and to disseminate it in these easy-to-use databases. So everything we do is either a database or a use case of our databases. And then another major goal of ours is to support design. So now that you know a little bit about who we are, I'm going to go around this figure and introduce each database. And then we're going to spend some extra time with a detailed discussion on MODULO, as well as a use case where that has been used to kind of advance basic science. And then a discussion of ALDB and a use case that sort of integrates MODULO and moves us more towards design. So I find it useful to sort of connect each of these databases to where they fall in the central dogma of biology. I'm going to kind of assume you know basically what this is. And we're going to begin by looking at our DNA databases. So ALDB is our first database, and this is one that I'm going to go into more detail later. But for now, I'll just say that this is where we store the mutations that we observe in our evolution studies. Our next DNA database is the strain database. And so the difference here is that these strains are wild, whereas the evolution strains were made by us. And so these might come from online public databases of known bacterial genomes, or they could be pulled right up out of the ground. We have a ton of soil samples as well as fecal samples. And this database basically stores the genomes of all of these strains and how the genes vary across different strains within the same species. And so each of these strains is also connected to the metabolites it can produce, the locations it was found in, and other phenotypes that the strain presents. Next up is the bitome. Now, this is a pretty unique and interesting DNA database where it's basically a giant table where each of the millions of columns are a single base pair of DNA, so a single letter moving down the genome. And then each row is a separate bit of information about that specific location on the genome. So for instance, is this letter an A? Yes or no? Does this protein bind there? Yes or no? And so with all of this binary information, you can actually train machine learning models to predict things like gene expression, and you can easily build a bitome for a new genome if you'd like. And so as we move towards design, we can create bitomes and use features from the bitome to predict things like how the promoter architecture should look. Next up is ProChIP-DB. And so this is the prokaryotic chromatin immunoprecipitation database. And this is where we store information linking proteins to the DNA sequences that they bind to. So some proteins are transcription factors, which will specifically link to certain sites on the genome. And that can give us clues as to how RNA expression works, how genes are generally regulated, and how we might want to design promoters again in the future. MODULO is the second database I'm going to cover later. But right now, I'll just say that these are machine learning results relating to this RNA level. So we finally moved fully out of DNA. And this is a very, very useful and interesting database for characterizing how cells regulate their gene expression. Next up is SSBioKB. So this is the structural systems biology knowledge base. And this is our protein database. This contains 3D protein structures, which are mapped to a ton of other knowledge and information about them, such as what reactions they catalyze, which different alleles exist across the pangenome, and which mutations commonly occur in different sites across the protein. And ultimately, this is going to be a really useful tool for mapping knowledge in 3D. Next up is big models. And this stores our metabolic models down here at the metabolite level. And so you can sort of think of a genome as encoding a bunch of different chemical reactions because it encodes different enzymes that catalyze those reactions. And so if you list out each reaction and then start to connect them in a map or a mathematical construct, you can actually see how different substrates are used in the cell, how food gets turned into growth, and what would happen if you were to introduce or delete a certain reaction. And so you can sort of look at how cells can be rewired or optimized by using these models. Finally, we have our two phenotypic databases, which are AmberDB and BiologDB. So that was a general overview, and now let's sort of step back to imodulons and what exactly is going on with our RNA database. And so with our approach, what we basically start with is a transcriptomic data set like this on the left, which shows the expression level of each gene across any conditions available to us. So these might be different food sources or stressors. And how these databases have historically been analyzed is one gene at a time. But we prefer to use kind of a machine learning big data approach and not deal with thousands of variables, but rather you allow a computer to group them together for us. And so we find co-regulated modules, which are called imodulons. And each of these contains a set of genes as well as a set of activity levels for each condition. And so looking at this example, we have the METJ imodulon, which contained a lot of genes that overlapped with what's called the METJ regulon. So this was a known, this red circle here was a known existing group of genes. And our imodulon recapitulated many of them. And then as we looked more into the genes that were excluded and included, we actually realized that the regulatory effects that were involved, namely this METJ binding, were more common, were fully captured by our imodulon and not necessarily by the existing regulon. So this can provide a new kind of top-down approach that might actually even be more accurate in some cases. And then if we look at the activity levels, we can look at this MAL-R imodulon, which regulates malate consumption. And so when you add malate to the media, this imodulon immediately activates. And so this entire group of genes turns on and it gets used to digest the malate. And then of course, when you add glucose to a malate media, the malate slowly turns off. And so what's important to know here is not only can we analyze these genes and activity levels, but we can do this all with big data alone. We don't need to do a ton of specific experiments, and we also don't need a ton of background information about the cell in order to do these computations and obtain some interesting results. So when we're done computing our imodulons and characterizing them, they end up getting placed on imodulon-DB. And so this is the splash page, and let's go ahead and travel to our original E. coli data set. So here you can see in this main table, all of the imodulons that were identified kind of listed out with some details about what they do and some statistics on them. And if we go to one of these imodulons, we have the RBSR imodulon here, which is probably regulated by the protein RBSR, which you can follow this link to learn more about. And then its genes are listed here. And interestingly enough, this is another case where there's an unknown or an unexpected gene mixed in. And so this is an opportunity for discovery here if this transporter might play a role with ribose. And then we have some additional gene plots. And then we can look at this activity profile. And you'll notice that one condition strongly activates these. And this condition here has the carbon source D2-deoxyribose. So when you feed the cell ribose, it activates ribose catabolism, which makes sense. And then, of course, there's some additional plots doing these comparisons with the known regulon. Now, our initial imodulon papers were very successful, so we decided to build out this project and do what's called the MODULO, where we mined all public prokaryotic transcriptomic data. And so a representation of that is here. So you can see this tiny segment here for corresponding to 1,000 data sets. And we looked at the entire set, everything that was published on this SRA database, and ultimately analyzed, downloaded, quality controlled, analyzed them for all of the most popular organisms, which are listed here. And so this covers model organisms, pathogens, intracellular pathogens, even in archaea. And currently we have posted 6 of these organisms on iModuLonDB, and there's 5 more coming soon. We already have 1,000 iModulons for you to browse on there, and 500 more on the way. So this is really exciting and provides a lot of opportunities to inform all kinds of research that might be related to the transcriptome. And so here's a basic science use case of iModulons. And here you can see the pure R iModulon, which was found in E. coli, and again had some of these genes that were sort of unexpectedly grouped together. And one of these genes was called YDHC, which at the time was just a putative transporter. And we know that pure R regulates purine metabolism. So there was the hypothesis that maybe YDHC is involved with that. And so after the work of this paper was done, really a phenomenal characterization going into how these genes, how this gene actually works. And it turns out to be an important purine transporter, and its regulator was also identified, which was originally YDHB, now PUNR. And the regulator responds to purines, just like pure R1. So there's kind of the same underlying signal controlling this iModulon. And then we were able to identify the underlying mechanisms for this particular gene and characterize and rename it. So moving on to our other and final database, this is the Adaptive Laboratory Evolution, or ALE database. And first you have to sort of understand what ALE is. And so what we do is take a starting strain and a condition that we're interested in evolving. And so this might be a stressful condition or something like that. We place the cells in this environment, and then we just give them plenty of time and room to grow. And what you'll notice over time is a few mutations will randomly pop up in these strains. And if the mutation is helpful to the cell, then that cell will grow faster and take over the flask. And then this process will continually repeat, and mutations will accumulate, and our fitness level will continually increase, meaning that this final cell is very well suited for the environment that we placed it in. And then we sequence the genome and obtain a set of mutations, which are the changes to that genome. And we have a good guess that some of these mutations will confer an actual fitness benefit to the cell. And so if we go to ALDB, we can see some general statistics about the size of ALDB here, and then we can travel to the experiments. We just click on the first experiment. SER stands for serine. And so this was a tolerization to serine. Apparently serine can be toxic to wild type cells. And so after evolving the cell in the presence of this stressor, we were able to see mutations arise. And you can look across the entire genome here. This is the genome on the x-axis, and then the number of mutations that occurred at a given site on the y-axis. And so these tallest bars indicate sites with mutations that are very likely to have given an actual benefit to the cell in surviving serine. And if you click on this mutations tab, you'll have access to all of this detailed data about exactly where these mutations occurred and which strains they happened in. So ALDB has been around for over 6 years, and its current size is 400,000 observed mutations across 16 organisms, 3,000 ALEs. And so the massive scale of this data set is very exciting, because it kind of opens up a lot of new opportunities for big data analysis and understanding how genomes change. And so here's one use case which kind of combines both of the databases that I talked about in detail. And so what we've done here is lined up the mutations from ALDB on the left and the imodulons that change as a result of these mutations on the right. And in the middle, we can begin to fill out some mechanisms and guesses and predictions about what might connect these mutations to these imodulons and the phenotypes that we observe. So for example, here we might have two mutations, and these are just generic ones, that might affect the TCA cycle, which might then lead into stressors like oxidative stress. And so if you're working on a genome design and you are noticing oxidative stress is becoming a problem for your cells, or you want to increase the flux through a certain pathway, then you might want to find these knowledge graphs and look through them and see which mutations might affect those phenotypes and imodulons of interest to you. And in this way, we can kind of build up a visual understanding of a strain in order to design better strains. So today, I introduced you to Genome Analytics team, where we learn from big data, and we've developed, to various levels, we've developed these 9 databases. We talked in detail about imodulon DB and how imodulons can inform which groups of genes are co-regulated and what their activity levels are, and how we've expanded that to cover so many new novel organisms. And then we also discussed ALDB, which has an enormous scale of mutations observed in evolutionary experiments. And so we really hope that genome analytics can have some impact on scientific discovery by sharing all of this information and by mining these databases ourselves for important knowledge. And then we also hope that this data can be used to engineer better strains to ultimately forward the goals of the CFB and just improve our lives in general. These are the papers that I used figures from, and I'd like to acknowledge the Novo Nordisk Foundation as well as these people who contributed to this presentation. So thank you.